a developmental cognitive neuroscientist from the University of Cambridge, and I'll be giving a presentation today describing some brain imaging data, which is from some participants from the ROOT cohort, which is a Cambridge-based longitudinal study of adolescent emotional development that I've been working on with colleagues, Professor Ian Goodyear and Dr. Tim Dalgleish from Cambridge. And we'll be talking about some tasks where we've been scanning participants, looking at how brain structure and brain function is affected by early life factors and genetic factors. Thank you. So, from the title, I'm going to talk about brain imaging, and I'm going to argue that this is a useful tool to help inform longitudinal studies, to tell us more information than we can understand just using longitudinal follow-up studies at the moment in order for understanding psychiatric disorders. So to qualify this, there's a couple of points I want to note from this title, just to clarify what I'm going to talk about. By psychiatric disorder, I mainly mean depression and anxiety disorders, although as is well known, there are significant comorbidities between other psychiatric disorders such as bipolar disorder and schizophrenia. There's also high degrees of depression and anxiety symptoms present within these other disorders. But in addition, I'd argue that all disorders are arguably related to impairment and self-regulation, and we can discuss this a bit more in the talk. And by follow-up studies, I'm not talking about performing longitudinal MRI studies, but rather using longitudinal data sets to inform neuroimaging studies. Okay, so I'm just going to briefly give an overview about what I mean by longitudinal epidemiological follow-up studies. And these are critical for understanding particular variables associated with disorders, such as which variables are antecedents, i.e. they come before the expression of the disorder, concomitants, they happen at the same time, or the final thing, consequences. By having the disorder, what happens then? So, and I'm going to use an example from a quite well-known psychological theory of depression by Peter Lewinson, who is a clinical psychologist from Oregon in the States, and he was one of the first people to argue and show using longitudinal studies that maybe the negative cognitions that were present in Beck's model of depression might not necessarily be antecedents, they might not predate a depressive episode, but rather maybe concomitants which express themselves when somebody is currently depressed. So I'm just going to go over his model just to give an illustration about antecedents, concomitants and consequences. So in terms of depression, it's well known that stressful negative life events, such as losing your job, predate an episode of depression. Okay, and in particular, so when someone suffers this negative life event, there is a consequence, a disruption in their daily routines, such as their habits. So say, for example, somebody loses their job, then there's a disruption about their whole life. So how are they going to pay their bills? How are they going to feed their family? Things like that. So all these automatic patterns of behaviour that are largely unconscious are subsequently disrupted by this negative life event. And so consequently there's a reduced rate of positive reinforcement. They're not able to do the activities that they enjoy, and there's an elevated rate of aversive experience. So they find the world more threatening. How are they going to pay their bills? How are they going to feed their family? The whole world is potentially more threatening. And then there's increased self-awareness. There's a focus on oneself. What did I do wrong? Why am I a bad person? All these negative expectancies, negative beliefs might then come to the fore. And then, as another in turn consequence, there's increased dysphoria and depression. This is present in the depressed state. And then there are consequences, and they may be behavioural. You may interact less with the world. There may be cognitive. You may show characteristic negative expectancies, even more emotional, somatic. You may feel greater number of somatic symptoms. Interpersonal, you may relate to others less. Okay, and in addition, underlying all this, there may be predisposing characteristics. There may be a genetic risk. There may be social risks, such as like a low social support group, or there may be immunities. There may be things that allow you to get out of this negative cycle and cause you to be immune to the negative life event. So I just wanted to, and also within the model there's feedback loops. So between The consequences may subsequently increase self-awareness, and there may be like a kindling effect for future episodes of depression. So the more one experiences negative life events, there may be increased risk for future negative life events, which then continue this cycle. So this model came out in 1985, and since then it's 2011. Things have changed with the invention of brain imaging techniques. And this is a slide from a hospital in Florida, where which seems the kind of thing they do, where they dress up their MRI scanner as a sandcastle, because it's thought this makes it more appealing to people having to go in there. And I just wanted to say how maybe ubiquitous in 2011 MRI scans are. And I wanted to ask the question, how many people have had an MRI scan? Just so I get an idea. Can you put your hands up if you've ever been inside an MRI scanner? So there's quite a few. So what I wanted to ask next is, when you went for an MRI scan, how many people had to fill out a questionnaire to say how they were feeling at the time of the scan? Was anybody here? Nobody did that. Not the scans you do in France. Okay, so how many people had a clinical interview? That was at the time of the scan. Did anybody to see if this was important, this was relevant? Nobody. Okay. And so if I was to say how many people had one of their parents interviewed to see if they could report any life events that happened in the first 10 years of your life, was this information collected at the time of the scan? So nobody. So the point I want to make is any of this information that's collected in this way relevant for understanding the pattern of brain activation that you get, or your brain structure? The answer is we don't know at present, because it's not been done. So the integration of epidemiological methods and cognitive neuroscience has been termed by others population neuroscience. And the aim of this is to fully understand the characterization of the human genome, the environment, pathological environments, or beneficial environments that we can grow up in, and also a refined characterization of the phenome. Okay, so the work I'm going to present today is essentially an integration of genetics. I'll talk about the serotonin transporter gene, epidemiology, and my interest is understanding the cognitive neuroscience, which allows us to understand the mechanisms that bridge genetics and epidemiology. Okay, so there's a number of different approaches to psychiatric genetic research, which I'm just going to give a quick overview about. So one is saying that genes essentially cause a disorder. And I'm not going to argue either way for any of these approaches, I'm just going to say them from the outset. Another is the endophenotype approach, where there's intermediate, could be neural aspects such as amygdala reactivity, or it could be other kind of neuropsychological test performance. The idea is that these measures are closer to the genotype than by understanding the disorder. Another approach is by saying, actually, rather than genes causing a disorder, it's the environment that causes the disorder, and the genes moderates the effect of the environment in causing the disorder. So this is a classical gene-environment interaction approach. And so what I'm going to talk about today is by employing this approach, that the environment causes the disorder and the genotype moderates it, and in fact we can measure the neural reactivity, which is a more proximate measure of these effects than by maybe a questionnaire or some other measure. Okay. And the main argument I'm going to talk about is that using certain tasks, we can measure how, in terms of reactivity, we can label it in this circumplex kind of graph where you have arousal on this axis and valence. So I'm going to say arousal, how we respond to environmental feedback, is largely genetically determined by the serotonin transporter gene. And I'm going to say that the valence, how we appraise that, so the feedback we get is either positive or negative, and this is environmentally mediated. And I'm going to unpack this more throughout the talk. Okay, so the main question I want to ask and answer, hopefully, with this talk is, how does the environment get under the skin? So we can understand, because genes are biological entities, we can understand quite clearly the biological expression of these genes, but we know less about how the environment negative environment gets under the skin. So there's that. And then secondly, how does genotype moderate the effects of the environment? i.e. how do genes and environments interact neurophysiologically that we can study with brain imaging? Okay, and as an example of a way to ask this question, I'm going to use data from the Roots cohort. And this is a longitudinal study of adolescent emotional development. And the PIs on this project, the principal investigators, are Ian Goodyear, who's a professor of child psychiatry in Cambridge, Tim Crowdace, who's a statistical psychologist, Valerie Dunn, who's a researcher interested in the effects of childhood adversity, Joe Herbert, who's a neuroscientist, particularly interested in the effects of hormones on the brain, and Peter Jones, who's professor of psychiatry. And I'm just going to give a bit of background about this cohort because, because this is the purpose of these kind of talks, and I want to set the scene a bit. So this is Cambridgeshire. This is a county which is to the north of London. It's about 45 minutes on the train. And participants for this cohort were recruited via schools in Cambridgeshire. There was 18 schools in total. So this is Cambridge here, and there was different schools to the north, south, east and west of Cambridge. And I'll just give an overview of the recruitment process. So we randomly recruited 14-year-olds to this cohort. Invitation letters and study information were sent to approximately 4,000 families via the schools, so we're able to get their names and addresses this way. And 1,238 gave written consent and entered the study. And then 1,185 students and 1,143 parents proceeded to the interview stage. And this is actually a staggering number. So both these students and parents underwent an interview at the initial part of the study. And the adolescents were interviewed in the school, and the parents were usually the mothers were interviewed predominantly in the family home. So this is a great undertaking, I'd argue. And I'll just quickly outline the theoretical framework underlying the Roots project. And so we were interested in distal vulnerabilities upon subsequent psychopathology. And these are things like genes, and there's candidate genes that we've studied. The usual suspects are serotonin transporter, BDNF, COMT, and glucocorticoid and mineralocorticoid receptor genes. And in addition, we were interested in the effect of the early environments and any adversities that occurred in preschool and middle childhood, and on the development of childhood temperament. And then there were proximal vulnerabilities that we were interested in studying, things like individual differences in morning cortisol and genetic sensitivity to endogenous cortisol and things like DHEA. And in addition, we studied psychosocial consequences, peer group difficulties, family difficulties, acute negative life events, and also the physical activities that the adolescents were undertaking. And then we looked at mood, cognitive processes, potential neural endophenotypes. So this is the work that I'll be presenting, but in addition, we had a neuropsychological test battery that was employed to measure things like emotional response, emotional recognition, decision making, and behavioural inhibition processes. And then finally, we want to know how do all these factors interact to give rise to differential risk patterns, which may underlie emotional, behavioural symptoms, anxiety, depression, conduct and oppositional disorders, and potentially psychotic symptoms. So this is an overview of the study. I'll just mention that there was a lot of different measures, different test batteries were employed. And so, like I said, participants came into the study aged 14, and then they were subsequently followed up 18 months after the initial study, and then again 36 months after. And we took reports of clinical symptoms, we did cognitive evaluations to measure things like rumination, self-esteem, lifestyle, eating, smoking, drinking habits that the adolescent engaged in. We conducted a structured clinical interview using the KSADS measure, and in addition took measures of physical state, height, weight, body, and like I mentioned, measures of cortisol and genetic information. So there's quite a lot of information gathered from these participants. And I'm going to focus on one thing in particular for this talk and talk a little bit more about that. And this is a measure of childhood adversity, what happened in the first 11 years of the child. So we use something called the Cambridge Early Experience Interview. And I'll talk through this. This assesses the quality of the family environment in the first 10 years of life. It's a semi-structured interview conducted with a child's primary caregiver, like I mentioned before, usually the mother. And it records the child's life experiences, the child's age of the occurrence of these experiences, both positive and negative, the duration, and there's an objective interviewer assessment of the impact of these experiences on the daily life of the family. And for the purposes of this talk, exposure to an adverse family environment was measured by ascertaining the amount of family discord experienced by the child. And this is defined as regular arguments, occasional physical violence, a lack of affectionate care or a lack of warmth or communication between family members. And in addition, the presence of any maltreatment, such as emotional, physical or sexual abuse, was also included in the definition. Okay, and because we did a longitudinal study, we were able to see if there were any potential reporting biases or whether the participants were consistent in what they reported throughout the different assessments. So, like I mentioned, participants underwent a structured clinical interview to ascertain if there was any psychiatric history. So, at age 14, 942 of the cohort did not report any psychiatric illness at any point previously. Okay. In contrast, 263 of the cohort did report any psychiatric illness. But the interesting thing is, if you follow these participants up when they're age 17, and you say, in the past, did you ever experience any psychiatric illness? Of those 263, 26% said, no, I've never experienced any psychiatric illness before. Which is an interesting finding. Is it why this occurred? Is it they just forgot that they'd experienced any psychiatric illness, or is it a reporting bias, or is it a methods problem? Maybe a different interviewer, we don't know. But it does actually concern me that this occurs, and also concerns me if in other studies where we recruit healthy volunteers and we say, has this ever happened to you? Then it might be underreported the prevalence of any psychiatric history. And it's a point which is very interesting to discuss. Okay, so I'm just going to mention retention in the study, and this might be of interest to other people performing cohort studies, how we manage this problem. And the secret, I think, is parents. Because these were adolescents, and by involving the parents in the cohort, it was very useful. So we'd call them up and say, hello, this is Nick from the Root Study. Can I ask you a few questions? And also because it's Cambridge, it's a university town, people are very interested in participating. So a lot of the parents were very keen, and so that minimised any dropout potentially in the study. I asked my colleague Valerie Dunn other aspects of what might have helped with retention. She said having a distinctive logo on the paperwork was also a factor. So if you'd like to do your own studies, then it's always good to get a nice logo. We also sent participants birthday and Christmas cards, and there was a termly newsletter, and we regularly updated a website with information about the study. Okay, so as I mentioned before, the specific problem that I've been interested in is understanding gene-environment interactions, and particularly what might happen in the brain which might underlie potential gene-environment interactions. So I thought it'd be good to just give a quick overview about this field, as I understand it. And it's a very fast-moving field, so there's new information coming out all the time which casts everything in a new light. So serotonin has been implicated hugely, in the literature involved in emotion processes, and I'm not going to say too much about that. But I'm not sure how many are familiar with the serotonin transporter gene. It's probably the most widely studied candidate gene in neuroscience, in psychiatry, and genetics, in terms of its potential effects on mental illness. So there's generally two variants. There's a short and a long allele. And so, as we know, serotonin is transferred by the synapse, and essentially, allelic variation, in psychological terms, modulates the efficiency by which serotonin is taken back up again and stored for future use. So it essentially varies, potentially the efficiency of the serotonergic system. And the first people to demonstrate an association between allelic variation and serotonin transporter with anxiety traits was Lesh in Science in 1996. So they, as I mentioned, they separated the groups into those with the LL allele and those carrying the S allele, so either the SS or SL alleles. And this is a measure of the reuptake of serotonin, and there was a difference in their in vivo investigation. And so they argued that the polymorphism has more of a dominant recessive than a codominant additive effects. And like I mentioned, they classed together the SS and SL participants into the S carrier group. I should mention here that in 2011, that a number of different variants have been identified, and it's a matter of debate whether this is still the best way to classify allelic variation. But this is what they did in this study. So they had the two groups, either the S carriers or the LL group, and they found an association between neuroticism and harm avoidance on questionnaires. So those carrying the S allele showed higher levels of neuroticism, anxiety-related traits than the LL group. And this is a classic finding. And subsequently, in a paper again in Science by Caspi et al., they found an association between the number of stressful life events that one experienced, and as I mentioned, it was moderated by the genotype. So those with the SS allele reported higher numbers of depressive symptoms than those with the other allelic variation. So this was the first evidence of a gene-environment interaction upon depressive symptoms. And so we want to know, as I mentioned, the underlying neurophysiology of these gene-environment interactions. And we know quite a lot about the underlying neuroanatomy of anxiety. And in particular, most researchers focus on this part of the brain called the amygdala. And it's been argued that in anxiety states, the amygdala is generally overactive compared to when you're in more of a calm, relaxed state. So the first person to examine whether allelic variation in the serotonin transporter gene was associated with greater amygdala reactivity was Ahmad Hariri in another paper in Science. And in this task, participants saw faces of different emotional valence, either angry or fear, or neutral faces, and they had to say which of these faces at the top showed the same emotion than those on the bottom. So in this case, it's this picture here. And then they had a control condition where participants had to say which of these shapes here was the same orientation as one of these on the bottom. Those carrying the S allele showed greater amygdala reactivity in the right amygdala compared to those in the L group. And so they did it in one cohort of 14 participants, and then they replicated it again in another one just to confirm the reliability of this finding. And then this was subsequently replicated in a larger cohort in a paper in 2005. But the interesting thing is in this study was that they found no relationship between anxiety traits measured with a harm avoidance questionnaire, the Cloninger questionnaire, and amygdala reactivity. So this was surprising because you would think that those with greater amygdala reactivity would show greater harm avoidance scores, but this wasn't the case from this study. And just to mention, the first study to try and demonstrate a gene-environment interaction was by Canley et al. And this was published in PNAS in 2006. And they found, interestingly, that those in the S group showed a decrease in amygdala reactivity with greater life stress, whereas the L group showed an increase in amygdala reactivity. And with hindsight, it's quite an interesting study. But one of the possible problems with it is they didn't cover for current state at the time of scanning. So we don't know if there's any effect of life stress over and above the emotion experience in the scanner. There's a number of other problems potentially, but I won't talk about them now. So since then, these were in 1996, and since then, what are the feelings in the field now about these results? So I'm just going to mention, well, a good way to bring together all the studies that have been looking at this problem since then is to perform a meta-analysis. And the most recent meta-analysis that I'm aware of, where they've tried to examine an association between anxiety-related traits and allelic variation in the serotonin transporter gene was in a paper that came out this year by Minelli et al. And they argued that a number of these studies did not employ a psychiatric interview at the time of assessment. So participants were just given the questionnaire and asked to fill out, and then they were genotyped. But when you screen participants for any previous psychiatric history and you exclude any of these participants, then there's no association between this gene and anxiety-related traits. So potentially this is like another warning of the importance of conducting a psychiatric screen at the time of assessment. And then the Caspi paper has been cited about 3,934 times the last time I checked, and this is a reliable finding. A huge number of studies, a number of times been cited, and it hasn't been replicated altogether. This study. It's worth discussing why this might be, and some have argued that the studies that did not replicate this finding were underpowered, and there was also inconsistencies in the analysis used. Some looked at and like I mentioned before, the way you characterise the groups has differed across studies. Some have included the LS group in with the LL group, and others include the SL group in with the SS group. And obviously this resulted in different results across studies. And there's a bit of a debate about what is the right thing to do, because it may be that there's a better model fit if you include the group in another group, but whether that is the thing to do is a matter of debate. And also some have argued that because of the lack of main effects this gene on depression, it means there cannot be an interaction. And again, this is a matter of debate. And in a review in 2008, giving a methodological critique of all the previous studies, Rudolf Uhr and Peter McGuffin at the Institute of Psychiatry in London reviewed 34 studies, and they found there were 17 full replications of this gene-environment interaction, there were 8 partial replications, and there were 9 non-replications. And the positive findings were more likely if an interview or objective assessment of the environment was used. But in contrast, large studies were more likely to use questionnaires. And then another meta-analysis that came out, the most recent one, looking at this, was by Karg et al. And they included 54 studies and a staggering number of over 40,000 individuals. And they found in their meta-analysis a significant gene-environment interaction. And then they performed a stratified analysis to see if the type of environmental stressor used moderated the ability to detect a significant difference. And in those that studied childhood maltreatment, they found this p-value. In contrast, those that studied stressful life events found less of a significant difference. And again, the method of measuring stress seems to matter. So those that use an objective measure had a strong p-value here, whereas those that use a self-report questionnaire tended to be less significant. So again, how you measure the early environment seems to be of critical importance. Okay, so I'm going to talk now about the brain imaging study that I've been working on for the past few years with colleagues, and I'll just give an overview of this study. So participants were recruited from the roots cohorts, and they were all identified if they had one of either the SS or LL versions of the serotonin transporter, and then or if they'd had childhood adversity using the Cambridge Early Experience Interview or not. And so I phoned them up and tried to recruit them, and then participants came for a pre-scan and screening questionnaire session, and then for one scan where we did these tasks looking at facial emotion processing, a cognitive control task, a feedback learning task, structural MRI, and then DTI scan. And then 2 weeks later they came back for a second scan where we looked at an emotion regulation task and a social evaluation task. And what I'm going to talk about today is the FACES task and the feedback learning task, and present some data from the structural MRI scan. And the interesting thing about longitudinal studies is actually how much data is collected by these studies. And so you might spend, say, 4 years actually collecting the data, and then you spend another 4 years analysing the data, because it's just huge. So this is the inclusion-exclusion criteria. As I said, they were either the SS or LL allele, normal corrected vision, they were white, so we tried to control for any ethnic differences, and English speaking. And the exclusion criteria is any current DSM axis 1 disorder, neurological trauma, participants were free from any medication, and we tried to control best for any IQ differences. So by having all the data, this wealth of data from the main cohort, we're able to compare the neuroimaging subsample with that of the root sample to see if it's representative. Oops. And we were able to recruit 70 participants of this sample, and those in the neuroimaging sample were slightly scored slightly low in depressive symptoms at the time of scanning, because we controlled, for any current DSM depression. There was no differences in the gender ratio, and there was no differences in the socio-economic status for those that participated in the neuroimaging task compared to the whole sample. Okay, so what I'm going to talk about for the data I'll present is the variables that I looked at in relation to brain structure and function. And in particular, the childhood adversity assessments, which was measured from 0 to 11 years of age. The psychiatric history assessment period, which was potentially in theory from 0 to 18. We did a life events assessment period, which measured life events a year before the last assessment, which was 36 months from the baseline. And then in addition, we have measures of current mood at the time of scanning. So I'm going to just quickly describe the participant information. So there was approximately 15, 16, 17, 18, 19 in each group. So allele CA plus means those with the allele allele and with the presence of childhood adversity. And then those without childhood adversity, SS, with childhood adversity, without. And there was no difference in gender across the groups, and importantly there was no difference in IQ. And often when people look at the effects of childhood adversity or trauma on brain structure and function, say for example, people have carried out studies looking at the effects of adoption on Romanian orphans in studies, those with institutional deprivation, the IQ tends to be significantly lower in these groups, which is potentially confounding the effects of early adversity. So yeah, those who had experienced early adversity had higher depressive symptoms at the time of scanning, but there was no difference in anxiety symptoms, and there was no differences in the negative life events which had occurred when measured a year before scanning. And in addition, those with adversity were significantly higher in that they'd had a greater number of psychiatric diagnoses prior to scanning. Which is nothing new, that if you've had childhood adversity you tend to have a greater incidence of psychiatric illness later in life. Okay, so as I mentioned, people were free of any psychiatric illness at the time of scanning, but because we had this data we were able to look back and say, did any of these participants ever report any psychiatric illness in one of their previous root assessments? And so 21 out of 70 had had a previous psychiatric diagnosis, and this included non-specific self-injury, anxiety disorder, an affective disorder, behavioural disorder, eating disorder, alcohol abuse and substance abuse. And some may argue this is a potential confound in our study that we included these participants, but it does also allow us to go back and see, does this variable impact on brain imaging, on brain structural function in quite a well-controlled way. So I'm just going to outline the measures of negative life experiences. This was a self-report questionnaire administered at age 17, and it asked participants to report unpleasant life experiences that happened to them, their family or closest friends over the preceding 12 months. And those life events were upsetting to the adolescents for more than 2 weeks. So again, this is different from other studies that have used things like a life event checklist, where they have to simply tick off if any of these life events had happened to them, because it had to have, in our study, significant impact for at least 2 weeks. And in addition, we measured current depressive anxiety symptoms with the Mood and Feelings questionnaire, a measure of depressive symptoms for this age group, and the Spielberg State Anxiety Inventory. And I'll just quickly go over some of the correlations. So childhood adversity was associated with greater depressive symptoms and previous psychiatric history. Those reporting recent negative life events also tended to have higher anxiety symptoms. Depressive symptoms was associated with higher anxiety symptoms. Depressive symptoms was associated with previous psychiatric history, but in fact genotype did not correlate with any measure at all. Okay, so I'm going to quickly go over some functional studies we used. One was a FACES task, And just to say in passing, a number of studies have argued that all these variables here are associated with greater amygdala reactivity. However, when people look at these variables, they don't tend to control for the other variables as well. But because in our study we were able to do this, we wanted to see whether, say, for example, people with the SS allele show greater amygdala reactivity after controlling for all these other variables. And this is the task we used. Participants saw faces and they were asked to identify the gender, say if the face they saw was a male or female face, and implicitly process the emotion. And they saw faces which are either sad, neutral valence, or angry valence. It's quite a straightforward task, commonly used in the literature. And the hypothesis was, do individuals at increased risk for depression show greater amygdala reactivity? And it's those with the SS genotype and in particular the childhood diversity groups. And this is, as I mentioned, after controlling for previous psychiatric history, current mood and recent negative life events. So I think the main contribution our study makes is to look at childhood diversity by taking into account all these other later variables, which hasn't been done so much previously. Okay. And we used a region of interest approach focusing on the left and right amygdala here, where the signal was extracted and then processed in SPSS. And to quickly go over the results, we found the main effective genotype in the left amygdala, with those with the SS allele demonstrating greater amygdala reactivity, not to any particular, regardless of the valence of the face. So they showed greater amygdala reactivity compared to the allele group, which replicated the previous Hariri paper. And in addition, we found no effect of childhood adversity upon amygdala reactivity, no effect of previous psychiatric illness, but we did find a significant contribution from recent negative life events. And plotting the data in this way for illustrative purposes, so this is the number of life events, so this is with no negative life events, or this is one or more, there was an additive effect. So those with the allele genotype showed no difference in, well, showed a big difference there in the SS group, irrespective of whether they experienced negative life events. But both groups showed an increase in amygdala activity, demonstrating an additive effect, not an interaction. So we did find the main effect of serotonin transporter gene, we found the main effect of recent negative life events, but we found no support for the effect of childhood adversity, which is in contrast to a recent report arguing that there's an effect of limbic scarring on the amygdala, that those with experienced child adversity measured with the child trauma questionnaire also demonstrate increased amygdala reactivity. And in addition, we found no support for gene-environment interaction hypothesis. But in this task, participants are kind of passively viewing the emotion. I think we need to understand more how the environmental feedback affects behaviour. And I'm going to, so like I said before, so the genotype, so those with the SS genotype may be more reactive to arousing stimuli, and it's an additive effect. But we're not looking at how people are able to respond, and so, but we may be simply increasing the activation of like a circuit looking at the punishment avoidance system or behavioural inhibition. But because we want to understand depression, which is what's been examined in these population level questionnaire studies, we're not able to do that with this task. So we need to employ maybe a different task to see how, when someone experiences behavioural inhibition, they may freeze and suppress the actual reward acquisition system, suppress exploratory behaviour. And so we need to come up with a task to look at this. So this is a feedback task. And I'm going to talk about a region of the brain called the habenula, which is a part of the brain which is becoming more prominent as we learn more about it and what it does. And it's a tiny part of the brain here. There's the lateral habenula and the medial habenula. And it's located at the top of the brain stem there. And it's important because it receives information from the limbic system, which may be to do with stress or arousal. And it causes a suppression of activity in the dopamine system and also the serotonin system. So the argument is, when one experiences either failure, punishment, non-reward, or pain, stress, it activates the habenula, which in turn suppresses the dopamine system or the serotonin system and causes motor suppression. And this may then decrease exploratory behaviour, approach behaviour, which is a common symptom in depression. So we wanted to use a task to see how people respond to external feedback from their environment. And the first study to look at this in healthy participants was by Ulsberger and von Kramer in Germany. And without going into the task in too much detail, they show that when one receives negative informative feedback, there's greater activation in this region here, which they call the habenula. And it's actually a very difficult part of the brain to scan because it's so small. But more and more studies have been performed looking at this. And they've argued that this region generates an error signal, which allows one to update their model of the world. So it's actually informative when one gets negative feedback from their environment, because they can then adjust their behavioural strategy and how they interact with the environment. So it's a negative prediction error signal, which allows a person to change how they their strategy for achieving rewards from their environment. So we wanted to use a similar task to the one that was used previously, maybe a simplified version of their task. And I'll quickly go over this task. Basically, participants saw cue letters on the screen, either A, B, C or D, and then they saw other letters, and they had to work out if these letters were associated based on the feedback that they got. Okay, so this is feedback. So I'll quickly show the task. And hopefully this will work okay. So they see a cue letter, here it's D, and then they see another letter, and they don't know if it goes together, so they have to use trial and error to begin, and they make a response and it says incorrect. So next time they see that pairing, they then need to learn that their last response was incorrect. So see another cue letter, this time A, and then they say this is incorrect, and by saying it's incorrect, they then got correct feedback, so this was the right thing to do. So then they're able to get essentially feedback from their external environment, which allows them to modify subsequent behaviour. Okay. So we did this task, and we found this region in the brain, which I'll argue was the habenula, was more activated by negative feedback than positive feedback. And then we wanted to see if any of the variables we had information on moderated the response to feedback. And we found that those who experienced negative life events showed a greater response to negative feedback and a diminished response to positive feedback. So what that means is when someone has recently had a negative life event, they tend to show an exaggerated response, maybe a catastrophizing response to this negative feedback, in contrast to those who did not experience negative life events. Okay, and it also affected task performance a little bit. So basically, prior negative life experiences moderate an individual's response to their subsequent feedback. And these negative life events increases an individual's response to negative feedback and decreases their response to positive feedback. And this may be a mechanism by which the environment may get under the skin. And it's important. So when participants, people come for fMRI studies, we should really ask them, has anything negative happened to you preceding the scan, because it may moderate their subsequent brain activation when they do these tasks. Okay, so the last thing I'm going to talk about is a structural study. So participants underwent high-resolution structural MRI, and it was used using a DARTAL, a standard imaging procedure. And we wanted to know if there was a presence of a gene-environment interaction upon brain structure, and in addition we wanted to see if there's any effects of recent negative life events or previous psychiatric history upon brain structure. And we didn't find any genetic effects, but we did find that those who had experienced childhood adversity showed reduced volume in a region here in the cerebellum, and also a small region in the medial prefrontal cortex. And then, in addition, we found no effect of recent negative life events upon brain structure, so it appears that these only tend to affect brain function. But we were able to match those participants with a previous history, which was 21, with those from our sample without a previous history, and we were able to match for genotype, gender and age. And we found that those who had experienced a previous psychiatric history showed smaller hippocampus volume in the left and right hippocampus. And we wanted to see, is it to do, so this is the effect here, that it's smaller in those with a previous psychiatric history. And we want to know, is it to do with any variables any information that we were able to collect throughout the study, is it to do with the type of the diagnosis that they had. So is it, and as you can see, there's no real difference here between those without. So it appears like there's all psychiatric illness, tends to show this based on our sample. Or is it to do with the number of diagnoses the participants had? So there was a number of comorbid diagnoses where participants had both anxiety disorder or both ADHD and anxiety disorder. And it seems, again, there was no effect of greater number of comorbid disorders. But as I mentioned, we covaried for gender because females tended to have a smaller hippocampus than the males. So what is the mechanism by which the cerebellum could be reduced by childhood diversity? We don't know. This is quite a new finding. But as I mentioned, the habenulus suppresses the basal ganglia, and as we know, you can inject tracers. This is work from monkey physiology, animal work, and shows that there are anatomical connections, reciprocal anatomical connections between the basal ganglia and the cerebellum. So the hypothesis that I'm going to put forward is that by suppressing motor behaviour, we suppress the function of the cerebellum, which may prevent the development of skills or adaptive motor behaviours, for interacting successfully with our environment. Okay, so in conclusion, I've tried to understand how does the environment get under the skin and how the gene-environment interactions arise neurophysiologically. And we found significant effects of the serotonin transporter gene upon amygdala reactivity. We found significant effect of recent negative life events upon both amygdala reactivity and the response to environmental feedback. We found an effect of childhood diversity upon cerebellum volume, and we found an effect of childhood history upon hippocampal volume. So without going into it too much, there may be a developmental, critical time window when these effects occur. So it may be in the formation of habits or skills which may cause the cerebellum to not develop properly. Whereas in contrast, we haven't looked at more conscious processes which may develop more through adolescence or later in life. So I can quickly sort of go back to the Lewinson model and say, what does the work that I've presented add to this model? Does it fill in any gaps about the underlying neural mechanisms? So the antecedents, the stressful negative life events, might be associated with habenular activation. The disruption of the scripted automatic behaviour plans by these negative life events might be associated with the basal ganglia and cerebellum. I haven't talked anything about changes in positive or negative reinforcement, but the increased self-awareness when one's in this hyper-aroused state as a consequence of the stressful life events may be associated with amygdala activation. And the consequences, having a psychiatric disorder may then cause changes in the hippocampus. And one predisposing characteristic that we've studied would be the serotonin transporter. And I won't talk about that. So there's a number of limitations of this work. So we've only focused on one gene, which is tiny considering how many other genes there are to look at. We need to examine other environmental factors, environmental pathogens. We've only performed a cross-sectional brain imaging study. We haven't followed people up over time to know if these brain activation findings are stable. And we don't know how our cohort relates to other samples, those with maybe neurodevelopmental behavioural disorders. And there's a great need to understand further the anatomical brain connectivity about how these brain regions are linked together. And I haven't said anything about gene-environment correlations, which is probably a whole different talk. There's a large number of people to thank, my collaborators in the University of Cambridge, in particular Ian Goodyear, who I mentioned, Valerie Dunn, the statistical psychologists Rosemary Abbott, Michelle St. Clair and Matthew Owens, who painstakingly collected all the data about recent negative life events and analysed that. Maria Ban, the geneticist we've worked with, Will and Lucy, who are two undergraduate placement students who helped me with the data collection, and other colleagues here involved in the writing and the conducting of the research. And also all the participants in the root study and the research assistants involved in this. And some cognitive neuroscientists that I've been collaborating with in Cambridge, Tim Dalglish in particular, who've informed the emotion processing side of the studies. And finally, the funders, who've been generous in their funding, and hopefully I've shown how brain imaging and epidemiological measures can complement each other. And I'll end with the only bit of French in the talk. Thank you.